When people ask me about CRISPR, they usually want to know three things. How does it work? What can we do with it? And should I be afraid? Since around 1970, scientists had known about a mysterious pattern that showed up in the DNA of nearly every bacteria. It looked roughly like this. One scientist told me that before CRISPR, he had to micro-inject roughly 1 million cells in order to get a single perfect mutation. With CRISPR, he could do the same thing using just 10 cells. The first human trials of CRISPR-based gene therapies are going to start this year for sickle cell anemia and beta thalassemia blood disorder, and also for retinal dystrophy, which causes blindness. One of CRISPR's biggest impacts will likely be in drug development. That's because pharmaceuticals are often designed to correct something that a gene is doing wrong, like making a protein that raises your cholesterol. Before CRISPR, it would have taken decades to genetically engineer and test each of these variants to see what effect they had. Now, using CRISPR, we can very quickly figure out what each variant does and which ones are important targets for medication. If you put an anti-malarial gene drive in just 1% of Anopheles mosquitoes, the species that transmits malaria, researchers estimate that it could spread to the entire population within a year. So in a year, at least hypothetically, you could virtually eliminate malaria. CRISPR is an extraordinary tool, and like all extraordinary tools, it has benefits and risks. Going forward, our most important job as citizens and leaders will be to understand these potential impacts and to discuss them with intelligence and clarity. Whether or not we manage to do that may literally determine our future.